Hello everyone, I'm Lisa and I'm a PhD student in the lab of Florian Schmidt. And in our lab we use nanobodies to study innate immune responses like the formation of inflammasomes, which is depicted over here. So upon infection with, for instance, a bacterium or a virus, inflammasomes get activated and also activate caspase 1. Activated caspase 1 can then cleave my protein of interest, which is gasdermin D. And upon cleavage, the N-terminus is released and can oligomerize into pores in the cellular membrane, which eventually leads to a very inflammatory type of cell death called pyroptosis. And how pyroptosis looks like in macrophages can be seen here, where compared to a blabbing apoptotic cell, a pyroptotic cell is a very blown up circular structure with a condensed nucleus. To study the process of gasdermin D pore formation, we immunized an camelid with full-length gasdermin D, which resulted in 6 nanobody hits. Of these hits, 3 actually seem to bind, according to the immunoassay, to the cytosolic gasdermin D. So we decided to continue with these 3 nanobodies. First, we wanted to look into the functional characteristics of the different nanobodies, and we looked into the release of LDH, which is an indicator for pyroptosis. Here we could see that nanobody 1 and nanobody 2 no longer showed the release of LDH. And also, when looking at IL-1 beta release, we could see that for nanobody 1 and 2, this is completely abrogated. When looking into the confocal microscope, we could see that upon stimulation, the presence of these red propydium iodide positive cells is reduced in nanobody 1 and nanobody 2 presence, which also indicates a reduction of pyroptosis. Instead, these cells actually undergo apoptosis, which is seen here, even though they still form specks, so basically an indicator for these inflammasomes. Then we wanted to know what the mechanism of inhibition was of these nanobodies, so we looked throughout the whole cascade, and first we could show that inflammasome activation was still intact in the presence of the inhibitory nanobodies, as was shown by the presence of, again, these inflammasomes in the flow cytometer. We could also see that cleavage of gasdermin D still happened, since the N-terminus could still be detected. Then we looked into the possibilities of oligomerization of gasdermin D, and we could see that under non-reducing conditions, there is no longer the formation of these higher order oligomers in presence of one of the inhibitory nanobodies. The last question we would like to address is whether or not the nanobodies still allow membrane localization of gasdermin D. This would allow us to answer the question whether oligomerization is needed for membrane insertion. So basically, if oligomers first form and then move towards the membrane, or that the monomers are first inserted into the membrane and oligomerized in the membrane itself. Anja from the Gaia lab has actually managed to crystallize gasdermin D in the presence of 2 nanobodies, which is depicted over here. This could nicely confirm our cellular findings. She also has a poster here, so please check it out. To summarize, we have found 6 nanobodies against gasdermin D, of which 3 function in the cytosol. Nanobody 1 and 2 inhibit pyroptosis via their interference with oligomerization, and we could see that these cells instead undergo apoptosis. So to answer our last question, whether oligomerization is needed for membrane insertion, we would like to perform some liposome assays to mimic the pore formation process. We would like to determine which caspases are involved in apoptosis, and we're looking into the functionality of the nanobodies in primary cells. Then in the meantime, we're still characterizing 16 new nanobodies to see if there are some interesting additional hits, and we are implementing them for microscopy, where we either add a fluorescently labeled nanobody to the outside of the cells, or we express them inside the cells with a fluorescent protein. If you have any questions, feel free to drop me an email, and thanks a lot for your attention.